In this lecture, we will discuss biochemistry of polysaccharides. You are watching my channel, Biochem Lecture. My name is Dr. Alam Zaid. I am a professor at the Department of Biochemistry, University of Malacca. I did PhD from the Institute of Biochemistry, Graz University of Technology, Graz, Austria. The contents of this presentation includes introduction to polysaccharide, classification of polysaccharide with examples, so first we will talk about the introduction of polysaccharide. Polysaccharide are also known as glycans. They contain many monosaccharide units which are joined together by glycosidic linkage. Sugar units may be more than 10, and some have hundreds or thousands of units. Polysaccharides are macromolecules with distinct properties. They may have amorphous or even insoluble in water. The sugar unit is mostly hexoses or their derivative, but they also include pentoses. Now, classifications. There are two main types of classification. Polysaccharide is classified into two main classes. One is known as homopolysaccharide. In homopolysaccharide, these compounds are made of only one type of monomeric residues or unit sugars. You can see here in this picture they may be branched or unbranched straight. And the second class of polysaccharide is heteropolysaccharide. A heteropolysaccharide consists of two or more different types of monosaccharides units which are glycosidically joined in different ways, like 1 to 4. In one case you can see here the different shapes. Polysaccharide can also be classified into three main classes. The first class is known as storage polysaccharide. In this class, those polysaccharides which have the function of storage. Now, in the second class is structural polysaccharide, which have structural properties. And the third class is functional, or sometimes called other, or sometimes called mixed polysaccharide. Now we will talk about the example. The first example of the polysaccharide is cellulose. Cellulose is a polymer of glucose which is found in plants. The highest quantity of cellulose is present in fibrous plant. It is important to know the mammalic beta-glycosidase enzyme which is responsible for the hydrolysis or the digestion. It is important structure component of the primary cell wall of green plants. It is made of a straight chain. It is usually a straight chain having no branch and is homopolysaccharide. You can see here one glucose is attached to another glucose by beta 1 to 4 glycosidic bonds. It is important to know that cellulose is the most abundant organic compound or organic polymer on Earth. Now some properties. The cellulose contains at the cotton fiber is very high, about 90%. That the wood is 40 to 50%, and that the dry hemp is approximately 50 to 60%. Cellulose is mainly used to produce paper boards and paper. It is important to note that ruminants and termites and even some birds, they can digest cellulose with the help of symbiotic microorganisms, especially bacteria. Cellulose is tasteless, it has no taste, it is odorless, it is hydrophilic in nature. In human nutrition, cellulose is a non-digestible constituent of insoluble dietary fiber, so it acts as a prebiotics. The next example is starch. Starch is made of glucose units. It is an example of homopolysaccharide and storage polysaccharide. Starch is present in all green plants, is a source of energy. Starch is white, tasteless, odorless powder. It is insoluble in cold water and alcohol. In this picture you can see there are two types of starch, one in the blue and one in the pink. So we can say that there are two types of starch, amylose and amylopectin. So the first type of starch, class of starch, is known as amylose. It is relatively insoluble starch. It is a polymer of D-glucose, in which the glucose is joined by alpha 1 to 4 glycosidic linkage. Amylose is the repeating unit, contain 300 to 3000 units. There is an enzyme known as alpha amylase, which digests amylose to maltotriose and maltose. You can see here in this picture they are joined to each other by 1 to 4 linkage. The 1 to 4 glycosidic structure promotes the formation of helical structure or helic. You can see here in this case the different colors represent the glucose moiety, and they form the polymer form a helical structure. Now function of the starch. Amylose, it is made up 20 to 30%. It is more resistant to digestion. Amylose is important in plant energy storage. It is more resistant to digestion than amylopectin. The digestive enzyme alpha amylase is responsible for its breakdown. We need to know this. It is epectoprebiotic. It is an important thickener, water binder, emulsion, stabilizer, and gelling agent. It forms starch iodine complex, which is used for its identification purposes. The next example is amylopectin. Amylopectin is water soluble fraction of starch. It is highly branched polymer of alpha D glucose unit. Branching usually occur between 24 to 30 units every. The branching occur at 1 to 6 bonds. It is stored in amyloblast. Amylopectin comprises of 70 to 80% of the starch by weight. It consists of around 2000 to 200,000 units. In this chemical structure you can see that in the straight chain the alpha D glucose is attached to each other by alpha 1 to 4 linkage, while you can see this branch point which is attached to each other by 1 to 6 linkage. Amylopectin is also used as a thickening agent or stabilizer. The amylopectin is hydrolyzed by amylase enzyme like amylose. The next example is glycogen. It is made up alpha D glucose units which are joined by alpha 1 to 4 glycosidic bond. Glycogen in glycogen, the branching occur usually at 8 to 12 sugar units by 1 to 6 linkage in the straight chain, while the branching it is 1 to 6. It is highly branched compared to starch, and it is present in animal. That's why it is called animal starch. You can see in this picture. The straight chain contain 1 to 4 alpha 1 to 4 linkage, while the branch part contain 1 to 6 linkage. It is structurally similar to amylopectin, but is highly branched. Glycogen is example of storage polysaccharide and homopolysaccharide. Glycogen is synthesized in the liver. It serves as a source of energy for the body. Glycogen in muscle, liver, and fat cell is stored in hydrated form along with the salt ion. Glycogen is non-osmotic molecule. It is break down to glucose for maintenance of blood glucose level or energy requirement. In this picture, you can see the microscopic slide at the liver of the animals. You can see this pink color. The area in pink is representing glycogen in the liver, which is surrounding the different cells. The next example of polysaccharide is chitin. It is a long chain polymer of N-acetyl glucosamine. The unit are joined by beta 1 to 4 linkage. It is a primary component of cell wall in fungi and exoskeleton of arthropods, in crustaceans, in insects, and the raduli of mollusks, and in fishes. It is also the important constituent of eye lenses, tendon, digestive and respiratory tracts, linings. In animals, it is the example of homopolysaccharide and structure polysaccharide. Sources of chitin includes those insects, crustaceans, the honeybees, and those mentioned in this picture. The major function of the chitin is formation of exoskeleton. They may be used as a fertilizer and for crop protection. In food industry, it is used as a thickener, thickening agent, stabilizer, and as an emulsion. Its derivative called chitosan is commonly used and has several functions. The next example is pectin. Pectin is a heteropolysaccharide. It is a linear polymer of galactouronic acid, sugar, and acetyl galactose. So it has different monomers. The structure also contains the galactose, arabinose, and xylose. The typical molecular weight ranges from 60,000 to 130,000 gram per mole, In nature, around 80% of the carboxylic group of galactouronic acid are esterified with methanol. So in this picture you can see one is carboxylic acid, another one is also galactouronic acid, and then acetyl galactouronic acid, and these are joined to each other to form a polymer. It is present in most of the fruits. It is used as a gelling agent, thickening agent, and stabilizer in fruits. The next example of polysaccharide is inulin. Inulin is a homopolymer, so we call it homopolysaccharide. It is made of fructose units. It is present in wheat, onion, bananas, garlic, asparagus, Jerusalem artichoke, and chicory. Chicory is a very nice source. You can see here the different units with fructose which are attached to each other, and they form a bond. The fructosyl units are joined by beta 1 to 2 bonds. It is used as a source of energy and for regulating cold resistance. It is also used as a dietary fiber. Inulin is used as for measuring the kidney function in human being.